So far, we've gotten a chance to look at linear electron flow in photosynthesis and the light reactions of photosynthesis. Let's very quickly review it with this simple sort of analogy. So comparing linear electron flow to a construction site. So remember that electrons that are taken from water when water is split sit here at the bottom of photosystem 2, and they wait for energy to be funneled towards them. This energy comes from light. So here you see a photon of light hitting this little electron and giving it a boost to the top of photosystem 2. Here, the construction worker will allow that electron that has gained the energy from the photon to sort of flow down this ATP mill. Right, and so this is very much like a hydroelectric turbine. The electron gives up some of the energy as it moves through the electron transport chain here. The electron transport chain will then make ATP shown here. At the bottom of the electron transport chain, the electron lands in the lap of photosystem 1. Here again, it waits for yet more energy to be absorbed from a photon. So here's a little photon that donates its energy to the electron to boost it all the way up here to the top of photosystem 1. From here, the construction worker will take that electron along with the energy that it's absorbed and give it to the electron carrier NADP+ to make NADPH, essentially dropping the electron in the NADPH bucket. So linear electron flow will use both photosystems, photosystem 2 and photosystem 1, and it makes both of the products of the light reaction, so both ATP and the NADPH, and it does split water to release oxygen to the atmosphere. Now, this is not the only way in which electrons can flow during photosynthesis. There are certain prokaryotic organisms and even some eukaryotic organisms that use cyclic electron flow to actually cycle electrons back from photosystem 1 through the long electron transport chain just to make ATP. So cyclic electron flow only uses one of the two photosystems, so typically only photosystem 1. And it produces ATP, but it does not produce any NADPH. That makes sense because if you're reusing the electrons, you can't really put them into the NADPH bucket and allow them to leave because you need to reuse them. On top of this, no water is split, so no oxygen is released as a result of cyclic electron flow. So let's take a look at what cyclic electron flow looks like in the context of linear flow. So in this particular slide, we have the full linear flow diagram, but some parts of linear flow have been grayed out. Those are the parts that are not used by cyclic electron flow. And so we're only going to be looking at the middle portion here that's actually colored. So during cyclic electron flow, an electron here, that special pair of chlorophyll A molecules at the bottom of photosystem 1, that electron will wait, absorb energy from the photons of light, and get excited up here to the primary electron acceptor. Now, instead of being handed off to the short electron transport chain to be put in the NADPH bucket, this electron instead will be carried here to the middle of the long electron transport chain that makes the ATP. And here it bounces down the electron transport chain, giving up some of its energy that can be used to make that ATP, and then lands right back where it started at that special pair of chlorophyll A molecules in photosystem 1 and waits for more energy to be funneled towards it, and it will just continue cycling over and over and over again. Now you might be thinking, well, what's the point? Why does cyclic electron flow exist if you're not making both products of the light reactions? And so we don't 100% know the answer to this particular question. It might be that cyclic electron flow is kind of an evolutionary relic. Many prokaryotic photosynthetic organisms will use cyclic electron flow, and so we think it's a simpler version of photosynthesis. It probably is somewhat similar to what evolved first when photosynthesis was first sort of an acquired trait of living organisms. On top of that, the few eukaryotic organisms that can do cyclic electron flow, we think that it might actually help with photoprotection. So when it's really bright outside, there's a lot of light, cyclic electron flow in some organisms that are photosynthetic might help to funnel away some of the excess energy so that the system doesn't get overwhelmed and molecules don't get damaged. All right, so now that we have a better understanding of how electrons will flow during the light reactions in photosynthesis, one more piece of the puzzle that we need to look at in a little bit more detail is how is exactly the ATP produced. I mentioned that the ATP that's produced during the light reactions is produced in a very similar manner to what you have in cellular respiration, nearly identical. And so what we're going to do in the next few slides is we're going to compare and contrast the way that ATP is made in the chloroplast during photosynthesis and in the mitochondrion during cellular respiration. Again, the process is pretty much identical with a few differences. One of the differences is where the energy comes from to make the ATP, and the other difference is how the electron transport chain, the ATP synthase, how all of those essential enzymes and proteins are actually spatially organized to perform this chemiosmosis that makes the ATP. All right, so let's take a look at energy first. In the mitochondria, the energy to make the ATP actually comes from chemical energy, right? The breakdown of glucose molecules. Whereas in the chloroplast, the energy to make ATP comes from light energy, from those photons that get absorbed by the photosystems. So if we were to now compare and contrast spatial organization, we're going to use this image twice, once on this slide, once on the next slide. So for this slide, where we're looking at spatial organization of chemiosmosis to make ATP in the mitochondrion, help me by only focusing on sort of the left-hand side of this figure. All right, so here's our mitochondrion. Remember, mitochondrion has two membranes. It has the outer smooth membrane and then the highly convoluted inner membrane. And then wherever you have the dark gray, this is where you have the high hydrogen ion concentration that's created by the electron transport chain. As the electrons move through the electron transport chain, they give up energy, and that energy is used to pump protons into that intermembrane space. All right, and so you can kind of see that here. Here's the inner membrane of the mitochondrion. Here's the electron transport chain. It pumps those protons into that dark gray intermembrane space. Remember, the protons, they don't want to be there. They want to flow back into the matrix, and they will do that, but only through the ATP synthase as they're flowing back into the matrix. And the ATP synthase then uses that exergonic flow of protons back into the matrix to make the ATP molecules. For the chloroplast, it's just a little bit different. Same principle, so we're still going to use the electron transport chain and the ATP synthase, but in terms of organization, things are a bit different. Just entertain me for a second. If we go back to the mitochondrion, right, and this blow up here, we're looking at this little cross section here through the cristae. Imagine that we took the cristae and we kind of disconnected it here, right, from the rest of the membrane. We would have a little dark gray bubble. And that's in fact exactly what we have here in the chloroplast, all these dark gray bubbles with the high hydrogen ion concentration on the inside. So now let's focus on the right-hand side of this figure. And now we're looking at this little blow up of a cross section through the thylakoid membrane. So here we have the thylakoid membrane. This is where the electron transport chain is and the ATP synthase. And here the electron transport chain, as the electrons move through it, protons are pumped from the stroma out here in light gray. So the stroma out here, they're pumped from the stroma into the inside of the thylakoid space, inside the thylakoid. So you can see the high hydrogen ion concentration here in the dark gray. From here, the protons, they don't want to be there, right? They want to flow back out into the stroma, and so they're only allowed to do so here through the ATP synthase, spinning the turbine and allowing the ATP synthase to generate a lot of ATP during the light reactions. Okay, so let's take the light reactions and everything we've learned with light reactions with cyclic electron flow, and let's put it all together in the context of the thylakoid. So in this particular image, we've got our thylakoid membrane, right? So one of those poker chips. And the inside here is the thylakoid space, and on the outside here in the light gray is the stroma. So if we start at the very beginning, water is split. The hydrogens are saved because they're a good source of electrons, and the oxygen is then released back to the atmosphere. The electrons from the hydrogen here are funneled in one at a time to replace electrons lost at that special pair of chlorophyll A molecules, P680 in photosystem 2. Here the electrons are going to wait. They're going to sit there and they're going to wait for the light harvesting complexes to funnel that energy from the photons into the reaction center complex in the middle. There, the electrons from that special pair of chlorophyll A molecules will absorb the energy and get boosted up here to the primary electron acceptor. Right? First redox reaction here. From here, the electron, along with its energy, gets handed off down this electron transport chain. As the electron moves down the electron transport chain, it gives up some of its energy, and that energy is then used to pump protons from the stroma out here into the thylakoid space, creating a high hydrogen ion concentration on the inside. So from here, the electron at the bottom of the electron transport chain that makes ATP will land here in the lap of that special pair of chlorophyll A molecules at the bottom of photosystem 1. And again, here the electron waits at P700 for the light harvesting complexes of photosystem 1 to funnel in that light energy to the center, to the reaction center complex. The electrons will absorb that energy and will boost it up there to the primary electron acceptor. From here, the electrons get handed off down a short electron transport chain, and they get attached to or given to the electron carrier NADP+ to make NADPH. And the NADPH that's created is created out here in the stroma. So the electrons are essentially complete. They're done for the light reactions. We have to still make the ATP though, right? So let's go take a half step back here. Remember, we have created a high hydrogen ion concentration in the inside of this thylakoid here. The protons, they don't want to stay there. They want to flow back out to the stroma. One place where they can do this is through the ATP synthase. As the protons flow through the ATP synthase, they spin the turbine, and that allows the ATP synthase to take ADP, link it with an inorganic phosphate to make ATP, again out here in the stroma. Both products of the light reactions, NADPH and ATP, are made out in the stroma of the chloroplast, which is a really good thing because this is exactly where the Calvin cycle takes place that utilizes both the NADPH and the ATP.